you have to draw an housefly on a plain sheet of paper, abi? And that housefly is very small, abi? Will you draw it that way, like this? No. It will not be clear. It will not be bold, abi? And that is not a diagram. Therefore, you are going to make use of what you call magnification. In that essence, you are going to enlarge, abi? You will not reduce it. Because the actual image is smaller, abi? We have to enlarge it, and you have to indicate your scale on the diagram. Is that clear? That's why I said that you are able to calculate this object that you have drawn here. For example, now, I'm now drawing my outside as big as this. As big as this. You can see it's too big, abi? Because this outside is small, abi? Therefore, I will make sure that I indicate the scale I use to draw it. I will now say mg is equal to 10 times, how many times bigger is this diagram as compared to this? How many times bigger it is? If it's 17 times bigger, 10 raised to power 17. Is that clear? If it's 8 times bigger, 8 raised to power 17. Is it clear? It means that you are going to calculate the size of this. If I draw, if all this thing in diameter is 80, abi, I will divide it, and I will now see how many of this will I get there. Is that clear? If I can get, for example now, how many 10 naira will I get in 1,000? How many 10 naira will I get in 1,000? Is that not 100? Very good, abi? It means that if I now say 10 raised by 100, what does it mean? 1,000. Abi? Do you get it now, abi? It means that I have used that scale to represent it. That's what I'm saying. And the same thing goes down to what? To reduction. If you have to draw an image in the lab, you can still reduce it. If you are you have to draw a monkey now, or to draw an elephant, which cannot be drawn the actual size on the paper, what do? You do reduction. So if you have drawn a very small image, I will now calculate the actual image, and I say this thing I've drawn here is minus raised to power the actual size. Don't worry. In our next class, you get it straight. You do it practically. You bring ruler, you bring your rope, and you're going to reduce, you're going to magnify. Is that clear? Meanwhile, go back to your GDP, your AI tools I give to you, and go and read on magnification and reduction. How to report magnification and reduction in what? In Barrel diagram. You must know the exact magnification and the exact reduction. It's very important for you to do that. Is it clear now? Is that clear now? And make sure that you always draw what you see, not what you're supposed to see. Draw what you see, not what you're supposed to see. Use an HP pencil. Don't sketch. Abi, make sure you draw the diagram on the right side and label on the left side. Don't use what? Don't shade. Can always dots to indicate a darker region. And make sure the diagram is as simple as possible. We are not fine artists. We are biologists. What you need to do is to just draw a simple image and show the details in it. Is it clear? And you can learn your diagram by what? By labeling. Hello, you can learn diagram what? By labeling. If you know the label of a diagram, you can draw it. For example, now, if you know that to draw a human being, require the nose, require the eye, require the mouth, require the ear, require the ear. If you know those parts, when you are drawing, you put them there. Abi? Definitely, if you know the parts of labeling of the animal, you can use the labeling to draw that animal. Don't cram the animal or don't cram the image in your brain. You are drawing a plant cell. Note all the elements of a plant cell. Know their position. Know their location. Those elements of a plant cell, the constituent of a plant cell, which is the cell wall, the cell body, the Golgi bodies, the nucleus, cell membrane, cytoplasm. If you know where they are, you can now be drawing it and put them where they should be. By that, you are okay. Don't cram diagram. Don't cram shape. Once you know the location of those labels of those parts, they will form your diagrams. Is it clear enough? Any question, please? Any question? If that is clear enough, that's how to draw in biology. And those are the rules and regulations gathering diagram drawing work in biology. We are not fine artists, I've told you. You will draw from now till next 3 years. You always draw. Even your theory classes. They will say, with the aid of the diagram, Abi, discuss. With the aid of the diagram, define. With the aid of the diagram, explain. You'll be doing that constantly. And they expect us from now to teach you that how to draw and rules of drawing. It will help you in practical, also help you in theory classes. Because in biology, you don't just mark. There are details that you mark in biology. For example, now, you draw anything, you don't title it. It's zero. You draw any diagram in biology, and you don't title it. No matter how good it is, it's what? It's a beautiful zero. Do you know why? You draw something like this, you label, I'll ask you, what is this one? Is this a goat, a cell, a monkey, I don't know. I cannot be thinking for you. The examiner will not think for you. It's you that will say, I have drawn a lion. Abi? And you now say diagram of a lion. Abi? Definitely, if it's the, I'll now look at it as a lion. Even though it's not a lion, I will look at it as a lion. Because what you have told me that you have drawn a lion, that will guide me how to mark. Okay, a lion has this, a lion has that, a lion has this, and I'll not start marking for you. Is it clear? But if title is not there, and you are telling me that you have done rubbish, and you have done jargons. And that is why it is good to title your what? Your diagram. Again, we have told you that it is good to draw on the right side, on the left side, and label on what? On the right side. Don't cross label. Do you know what cross labeling? For example, now, if you label this way, another one this way, this is an intersection. Abi, it's a cross labeling. Make sure your diagrams are labeled separately without any intersection. For example, now, if you look at you can look at that very way. You not say labeling here that it's accepted. But when you are drawing your own, lines will be cutting each other. Is that clear? Just because, for example, now, seed bank, seed leaf. I mean, seed one is rubbish. Yeah, it has cancelled this, that's cancelled this, that's cancelled that. So 3 diagrams are wrong, 3 labels are wrong already because you have already crossed them out. Is it clear? The next thing that make sure you pluralize or use singular where necessary. Make sure you pluralize or use singular. For example, now you draw this one. You now say eyes. And you're only touching only one eye. Is that eyes? Is that clear? We have flagellum, flagella. We have cilia, cilium. Is that clear? When you say when you say cilia, it's only one. It's many. When you say cilium, you see, when they are in plural, you never know. And in singular, it does know. If I label 2 things like this, and I say eye, is it one high I pointed to? 2 high. And I say high, is 0. Because I don't say eyes. Is it clear now? Because I have taught 2 parts. So singular and plural should be used where necessary. Whatever you touch, and make sure your diagram, your labeling is touching the right part. Hello? Your what? Your labeling is what? Is touching the what? The right part. For example now, this is the embryo. Here. Abi? And I now touch here. And I say embryo. See the space, Abi? Between the embryo and your line. So can you go down? So you are drawing, you are pointing to something else. I can think it's a cytoplasm. Because it's not touching the embryo per se. Is that clear? If you don't clear enough, if you don't come back enough, can you be enough? Is that clear? You talk Tibo, because of your lab of a can or your can Dubai or any high. I will say no. Is the Irish, is the hide. Or go to be Dubai on your high brown. You tell label here. Is it high brown? Is that clear? You know, it's Stephen on the Obosian on the nose tree. It's not nose lele. You know, you won't see those three. The passage. Is it clear? So you make sure that your diagram and your labeling touch the right part. All this I'm giving to you are how we mark in biology diagrams. Is that clear? We mark what? Magnification. Abi, if you draw something, you don't put NG there. You have drawn. I don't know what you have drawn. I will give you -1 there already because I don't know how big your image is. Is that clear? I don't know how small it is. But if you tell me magnification, I will not even check. I will not even go and take ruler and start checking if it's actually long or it's short. Once you are reporting to me that it's times 10, it's about 3, I will assume you are correct. Is it clear? You title your work, and title of your diagram must always be biology recommended that it should be at the top. Is it clear? At the top, not below. Do you know why? If I draw mango here, I draw orange here. Is that clear? And I say an orange. Here, Abi, an orange here. Which one am I referring to? Is it the one up or the one down? Is it clear? Or by the time I say a mango, a orange, I will know that this one that's here now is referring to the one down. Abi? So I will assume that the title up is referring to the something down. And be sure you are drawing whatever you draw. Maybe you draw the ventral view, the dorsal view. What's a ventral? Ventral means from here. This is ventral now. Is that clear? This is ventral, this is dorsal. If you draw from the back, maybe if you want to draw me from ventral now, you draw from my stomach. Is that clear? You want to draw from dorsal, you draw from my head, down. Do you get it now? You can draw horizontally or vertically. Can I say the ventral view of an heart, the ventral view of a lizard, the dorsal view of a fish? Is that clear? The view that you draw. Is that clear? We still go through that. We still go through the views. The view means the angle that you are using to view that image. Is that clear? That angle from the back, from the side, from the top, or beneath. Is that clear? Can be Hana, can be Doza, can be Ventra. We see to that. So you know the proper labelings, not just a mango. Which view are you drawing? Is it clear now? It's also important as well. All these that I've taught you now are things that you should take care of. Are you good to go now? Are you good to go now? You are good to go in diagrams. You now know that in biology, diagram is not complex. Abi? You now know that whether you are a female, you are a male, diagram should not give you phobia. Do you agree? Because I have told you in the class now that in biology diagram, be simple as much as possible. Abi? So no need of going to contractor. Do you know what a contractor? Contractor is somebody that will be in your class like a guy that can be drawing. Because I've told you already that it is be simple as possible. Is that clear? And the diagram itself cannot fetch you a good mark. Like the magnification, the title, the labeling, and the ruler use. Abi? This actual image itself is not as, the mark is gathering, it's not as much as other parts you can draw your own. Therefore, erase the phobia in your mind and make sure that you draw your own henceforth. Is it clear now? Is it clear? You now promise me that all of you will take charge of your image and make sure that you draw on your own henceforth and you don't take it out again. Because all the rules I've told you now, you can follow the rules, forget your mark. You're okay. Forget issues with biological diagrams. You'll be okay. Are you okay now? But any question, please? Any question? Any question now? I guess you are a brilliant student.